Hi guys, Jason here, and welcome to Med School Hero. So today we're going to look at the biochemistry of cancer. Now, the word cancer in itself comes from the Latin word cancrum or Greek word carcinoma, and both words mean crab. The reason why they call it crab is because of the way swollen veins around the tumor resemble a crab's legs. Now, in itself, cancer simply means uncontrolled cell growth, where a single cell will acquire mutations, refuse to stop dividing, and produce a mass we call a tumor. Now, why is this topic so important in biochemistry? Well, for one thing, cancer is the second leading cause of death worldwide, and also in exams, questions on the etiology of cancer, on oncogenes, on tumor markers, and on some treatment principles are often high yield. So, in the next couple of minutes, we're going to break it down into simple, structured, and exam ready bits. Properties of cancer cells. But first, let's try to understand what makes a tumor benign versus malignant. Benign tumors grow slowly. They are usually encapsulated, which means they stay inside a fibrous capsule and they do not spread to other tissues or organs. They compress surrounding tissues rather than invade it. And most importantly, benign tumors do not metastasize. They can still cause problems if they are in enclosed spaces like the skull, like in this example. But they are not as dangerous as malignant tumors. Malignant tumors grow rapidly, they invade other tissues, they are poorly differentiated, and they lack adhesion. And the most important of all for malignant tumors, in fact, what examiners want you to know and what you should know is the fact that malignant tumors carry out metastasis, which is the ability to spread to distant organs. In fact, metastasis is the hallmark of malignancy of cancer. Now, you can take a look at the diagram and appreciate the differences between benign tumor and malignant tumor, as you can see the encapsulation there. Then you can also see the table that is clearly highlighting the differences between the two concepts in an exam focused manner. Now, beyond this classification, cancer cells also show distinct morphological and biochemical properties. For one thing, morphologically, cancer cells are often pleomorphic and irregular in shape. They are larger and they have hyperchromatic nuclei. The way they grow is that they grow in multi layers, losing contact inhibition. Then biochemically, cancer cells show increased DNA and RNA synthesis and also increased aerobic glycolysis, to be precise. But the special thing about glycolysis in cancer cells is that even with or without oxygen, they're able to break glucose to lactate and lactate to ATP. And this is called the Warburg effect. They also show altered membrane glycoproteins and secretion of proteases and kinases. Now let's take a closer look at this Warburg effect, where glucose is broken down to lactate and ATP is generated from this. Now this is so unique because cancer cells do this with or without oxygen to generate the energy they need to keep growing and dividing. Etiology of cancer. All cancer have multifactorial cause and origin. Some of these factors are age, where there is increase in risk after the age of 55. There is also genetic mutations that can cause conditions like retinoblastoma and BRCA mutations. Then environmental and lifestyle factors like smoking, alcohol, and high fat diets are strongly implicated to causing cancer. Carcinogens, which are cancer causing agents, are grouped into three: physical agents, chemical carcinogens, and biological agents. For physical agents, radiation such as from X-rays and ultraviolet light cause DNA breaks and pyrimidine dimers. Ultraviolet light especially is the most potent, causing pyrimidine dimers, which are these kind of lesions or injury to the DNA that damage the DNA's pyrimidine bases. Then for chemical carcinogens, aflatoxin B1 in moldy granules cause hepatoma. Benzopyrene in cigarette smoke causes lung cancers. For biochemical agents, oncogenic viruses like EBV cause Burkitt's lymphoma and nasopharyngeal carcinoma. HBV 16 and 18 cause cervical cancer. HBV and HCV cause hepatocellular carcinoma. Take that in mind. Now, oncogenes and tumor suppressor genes. So now we are in the heart of biochemistry of cancer, which is genes. Proto-oncogenes are normal regulatory genes, but when they are mutated, amplified, or translocated in any way, they become oncogenes. These oncogenes, imagine it like hitting acceleration in your car to make the car just be moving fast. Those are what oncogenes are, out of control proto-oncogenes. And examples are here: ROAS, MYC, ERB, which is a growth factor receptor, SROC, which is a tyrosine kinase protein. So all these are normal proto-oncogenes, but when they're out of control, they become oncogenes. Now in this table, you will see examples of proto-oncogenes, their normal functions, and their oncogenic mutations. For the ROAS proto-oncogene, the normal function is signal transduction, but when it is mutated, it leads to uncontrolled cell proliferation. Then for the MYC proto-oncogene, the normal function is to regulate cell cycle genes, but when it is mutated, it leads to Burkitt's lymphoma. Then for ERBB gene, it's a growth factor receptor, and when it becomes translocated or mutated, it leads to breast and gastric cancers. Then SROC proto-oncogene has tyrosine kinase activity and is important in cell signaling. When it is mutated, it leads to uncontrolled growth as seen in sarcomas. So there's persistent tyrosine kinase activity. Now, tumor suppressor genes. These genes stop genes from going out of control. They are like the brakes to your car. When they are lost or inactivated, the cell will lose control. Examples: we have the P53, which is the guardian of the genome. It halts the cycle of DNA repair or pushes damaged cells into apoptosis. The RB gene prevents unchecked cell cycle progression as well. Then there's also the BRCA12 gene, which is linked with hereditary breast and ovarian cancer. So high yield exam correlation now. Loss of both alleles of that RB gene causes retinoblastoma, very important. Then mutated P53 is found in over 50% of human cancers. So remember this concept. Oncogenes are like hitting acceleration on your car, and tumor suppressor genes are like the brakes that prevent the genes from going out of control. Telomeres and telomerase. Cancer cells are virtually immortal. Why? One reason is the activity of the enzyme telomerase. But before that, you need to know that telomeres are repetitive DNA sequences located at the ends of chromosomes, as you can see here. Now, normal somatic cells lose telomere length every time they divide, just like a biological clock. So when it becomes short and short and short, they will not be able to divide again. But in cancer, telomerase is reactivated. The enzyme preserves telomeres and grants cells unlimited divisions. That is why telomerase is being explored as a therapeutic target and even as a tumor marker in certain cancers. Tumor markers in clinical practice. Tumor markers are proteins or molecules that are measurable in blood. They are used for monitoring, prognosis, and recurrence, but rarely used for diagnosis alone. Some examples are PSA, which is linked to prostate cancer, CA125, which is linked to ovarian cancer, beta HCG, which is linked to choriocarcinoma and germ cell tumors, then AFP, which is linked to hepatocellular carcinoma, and CEA protein, which is linked to colorectal cancer. So these are some examples of tumor markers. Angiogenesis and metastasis. To grow beyond a few millimeters, tumors need their own blood supply, so they will secrete angiogenic factors like VEGF and BFGF. Metastasis, which is the spread of these tumors to distant organs, is the ultimate proof of malignancy, and enzymes like collagenase and stromolysin help them to invade other tissues. So you can see it in this flow chart here. So you see the normal cell with the proto-oncogene acquires a mutation, the proto-oncogene becomes an oncogene, and a tumor develops, and angiogenic factors are secreted to create its own blood vessel so that it can carry out metastasis and invade other tissues with the help of enzymes like collagenase and stromolysin. Okay, so to recap in summary, we've learned that cancer arises from mutations triggered by mutagens, viruses, or lifestyle factors. 2. Oncogenes drive growth while tumor suppressor genes normally restrain it, like acceleration and brake. 3. Telomeres keep cancer cells immortal. 4. Tumor markers are clinically useful for monitoring, not really diagnosing alone. 5. The key feature of malignancy is metastasis. So that's it. Biochemistry of cancer in 11 minutes.
If you found this helpful, don't forget to like and subscribe. Let's study smarter and not harder.
Thanks for watching, and I'm just hoping to
See you in the next video.